Hi, my name is Jovita Jimena Villanueva Rosales, and today I'm going to talk about the cell. When we talk about the cell, we are talking about the basic structural and functional unit of living beings. Cells are complex and highly organized, whether an organism is made up of a single cell or many cells. These vital units have internal parts in a specific position, which act under fine regulations to prevent errors and allow their function to be carried out. They possess the ability to replicate. Cells replicate by division, a process in which the content of a mother cell are divided between two daughter cells, whereby the mother cell has duplicated the generic material so that the new cell receives a complete and identical set of the origin information. Cells also obtain energy to maintain the cell's high degree of organization and complexity and to sustain its metabolism. The cell needs a constant supply of energy. This energy source depends on the organism. Finally, cells interact with the environment. Cells possesses various structures on their surface which act as receptors through which various external agents generate specific responses in the cell. Now that we know that cells are the fundamental units of life on Earth and that they are the building blocks that make up all other living things, now we should know that every cell contains a set of organelles, subcellular structures that are especially adapted to carry out necessary functions of life. For example, animal cells contain numerous organelles to help them carry out the functions essential to their survival. It has a nucleus, a mitochondria, a nucleolus, a lysosomes, a microvilli, a chromatin, nuclear membrane, centrioles, microtubules, cytoplasm, Golgi complex, ribosome, a smooth endoplasmic reticulum, broad endoplasmic reticulum, and plasma membrane. Plant cells contains all of the same organelles as the cells, including mitochondria and nucleus, ribosome, smooth and rough Golgi apparatus, lysosomes, peroxisomes, cytoplasm, and cell membrane. However, they also contain some subcellular structures that are absent in animal cells, such as chloroplast, a vacuole, and a cell wall.
Hi, my name is Judith, and I will be talking about the characteristic of living things and the difference between eukaryotic and prokaryotic cells. The first characteristic of a living thing is that they must have at least one cell. The second is that they have a metabolism, and metabolism is the chemical reaction to sustain life. The third one is homeostasis, which is the maintenance of internal conditions. The fourth one is a response to stimuli, which is when they respond to the environment. The fifth one is that they must have DNA. The sixth one is that they must have the ability to grow. The seventh is the ability to reproduce, sexually or asexually. And the final one is evolve, which means that they adapt to the environment to survive. Now I will talk about the difference between the prokaryotic and eukaryotic cells. To start this, we should know that the eukaryotic cell is more complex than the prokaryotic, which means that the eukaryotic has more things. The prokaryotic and eukaryotic cell both have cell plasma, membrane, cytoplasms, DNA, and ribosomes. Eukaryotic cell has mitochondria, centrioles, Golgi complex, chromatin, microvilli, and lysosomes, which are some of the characteristics that prokaryotic cells do not have.
Good afternoon, my name is Josgar Acosta, and today I'm going to be talking about the history of the microscope. The first compound microscope, which is a microscope of two or more lenses, was created by Janssen, who created a compound microscope of two lenses, which has a magnitude from 3 to 9 and has one lens at the top and one at the bottom of a tube of 50 centimeters. The second one was created by Robert Hooke, who created a compound microscope of 3 lenses and 4 tubes, which has a magnitude from 20 to 50. And the last one was created by Anton van Leeuwenhoek, who created a compound microscope with spherical lenses, which allowed the compound microscope have a magnitude from 200 to 300. And also he was the one who perfected the simple microscope with the same spherical lenses. Now I'm going to explain a bit about the cell theory. This theory proposed that every living being is composed by cells, no matter their differences. And this theory is based on 5 postulates. That the first one says that all living beings are made of cells. The second one says that the cell is the physiological material of all living beings. The third one says that the cell comes from a pre-existing cell. The fourth one explains that all cells have DNA or genetic material. And the most recent one, and the fifth one, explains that disease starts from the cell. All these postulates are based on the observations of Robert Hooke and Anton van Leeuwenhoek with the original composed microscopes.
For a cell to stay alive, it must exchange material with its environment and to communicate with other cells through the membrane. For this to happen, there are 2 types of transport: the passive one, that doesn't require an energy, and the active, that requires an energy. In the passive transport, there is a diffusion, in which the molecule moves from a higher area of concentration to a lower area of concentration. The osmosis has the same process of diffusion, but it is only for water and needs to pass through a semi-permeable membrane. Facilitated dissemination is similar to simple diffusion, but it only allows protein to pass through the transport protein and the channel protein. Carrier proteins, also called transporter proteins, change its shape to enable a molecule to pass from one side to another, so when the molecule has passed, it comes back to its original shape. Channel proteins allow molecules to move through the membrane by acting as a gate that opens or closes according to its demand. On the other hand, active transportation is when a carrier protein moves a molecule against a concentration gradient. What does that mean? Is that is the same thing as diffusion, similar, but it moves a molecule from a lower concentration to a higher concentration. In the endocytosis, the cell is engulfed by the plasma membrane and transported through the vesicles. There are 3 types. First, the pinocytosis, where the plasma membrane forms a dimple and it only absorbs liquid. The phagocytosis is the same process that pinocytosis. The only difference is that it forms the pseudopods that envelop the solid bacteria. The receptor-mediated endocytosis has these receptors, as its name says, that engulf certain bacteria. The endocytosis, however, doesn't absorb material, it releases material.
Hello, my name is Ana Arevalo, and today I'm going to be talking about the topics of levels of organization and homeostasis. Let's start with levels of organization. So, all bodily functions perform a task simultaneously, and the organization of the parts of the organism are really important to understand how an organism works, and the level of each of its components, the smallest to the largest. In this organization, we start with the atom, which is the submicroscopic level that makes up life, and these same atoms, when intertwined, create molecules, the smallest particle of a compound with chemical properties. Molecules would form organelles that build the parts of a cell, which is the fundamental unit of life. Cells with similar functions and structures performing a task together are known as tissues, which are the building blocks of organs. And when there is a group of organs carrying out a process, it is a system. For example, the digestive system. And all these elements create an organism. Now let's go with homeostasis. Homeostasis is a physiological system with the ability to maintain internal conditions by measuring and compensating for changes of the outside world that could damage them. It constantly regulates body levels of acid, blood sugar, blood pressure, among others. The first person to suggest that living things keep their inner functions constant was Claude Bernard in the 19th century. But it wasn't until 1929 that Walter Cannon named these regulatory mechanisms. But how does the body maintain homeostasis? Since there are external and internal factors that cause disturbance, homeostasis uses positive and negative feedback to counteract or intensify changes. The negative feedback is when the system responds to stimuli by regulating or counterattacking it. For example, if the body temperature is too high, this feedback loop will try to bring it back down. And positive feedback is when the system moves from its initial state and enhances or amplifies the stimuli that produce the change, seeking to reinforce it. An example is childbirth, in where the cervix, which is usually closed, opens for the baby to come out. With both feedbacks, the organism keeps itself regulated and in most cases, safe. Thank you for your attention.
Hello everyone, my name is Angie and today I'm going to talk about 3 different topics. The first topic I'm going to talk about is pregnancy. As we know, pregnancy is the process of a fetus developing in a woman's uterus, that this process usually takes just over 9 months. There are different stages of pregnancy: the first trimester, the second trimester, and the third trimester. The first trimester is the first 12 weeks when conception occurs and the fertilized eggs implants on the uterine wall. The second trimester, weeks that are from the 13th to the 28th, when the fetus develops and the mother may feel movements. And the third trimester is the final stage of the pregnancy. Now I'm going to talk about addictions. Addictions may be on social networks, like every social media, because now we adolescents used to have like our life in social networks because we have like this dependence, and also we used to get influence about groups and when they ask us like, do you want drugs or something like that, we usually say yes because of the pressure of the moment and to get more friends. Now I'm going to talk about obesity. The obesity here in Mexico is too much common than in other places. And this is something we usually do not we can control as much as we wanted to, because this is more like in the person, he or she needs to change their mind. Also, now in the adolescence we have too much anxiety, and we think the fast food will cure this anxiety, and we usually eat too much to feel better with ourselves.